Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, I will discuss a very important topic with you people, and the title of my today's lecture is Bacterial Cell Wall Inhibitors and the Mechanism of Action of Different Antibiotics. And those antibiotics are basically the cell wall inhibitors. Okay, so in today's video, we will discuss that how the peptidoglycan layer of a bacterial cell wall is degraded by cell wall inhibitors by different antibiotics that are penicillins, vancomycins, and cephalosporins, and monobactam, epinephrine. These all are the antibiotics that are used against the bacteria in order to inhibit their peptide, in order to inhibit their growth, and in order to kill them, and stops their biosynthesis of the peptidoglycan layer. In the previous videos, I have discussed that how bacteria, what is the basic structure of the peptidoglycan layer, and and I have also discussed the biosynthesis of the peptidoglycan layer. Okay, so I recommend you to watch that videos, because in order to provide a background for yourself, first watch that video, so then you can easily understand that how antibiotics, how the cell wall inhibitors acts on the bacterial cell walls, the peptidoglycan layer, and destroy their structure and inhibit their growth. Okay, so let's start with the video lecture. First of all, you should know about the peptidoglycan layer, that where it is present in the gram-positive bacteria. So it is the outermost layer, or that is present in the gram-positive bacteria, and it is very much thick. It is thick because it consists of 45 to 50 sheets of peptidoglycan. Okay, and this is first number sheet, this is second number sheet, this is third number sheets. Okay, so and it is about 45 to 50 sheets. Okay, and keep this thing in mind, that peptidoglycan layer is very much important for the survival of bacteria, because it provides integrity, it provides strength to the bacteria, and it provides strength, and bacteria, with the help of this peptidoglycan layer, bacteria can withstand different adverse conditions. Okay, so if we destroy this peptidoglycan layer, so the bacteria become very much weak, and it can be easily degraded by osmotic pressures and different other external adverse conditions. Okay, so keep this thing in your mind. So this is the gram positive bacteria, the cell wall of gram positive bacteria, and this is the peptidoglycan layer that is present in the gram negative bacteria. Okay, this is the peptidoglycan layer, it lies next to the outer membrane. Okay, so just for your information, gram negative bacteria are a bit resistant to antibiotic. I don't mean that it is totally resistant, and but with respect to gram positive bacteria, gram negative bacteria are hard to kill, because it consists of an outer membrane. After that, it consists of an inner membrane, this peptidoglycan layer. So cell wall inhibitors are mostly acts against gram positive bacteria. It also acts against gram negative bacteria. I'm not saying about that it only acts against gram positive bacteria, but gram positive bacteria can be easily killed by the cell wall inhibitors, because the outermost layer is the peptidoglycan layer. Okay, so the antibiotics can get easily access to that layer. So that's why cell wall inhibitors are best against gram positive bacteria. Okay, so keep this concept in your mind. Now here come the real action of antibiotics. Okay, this is basically the biosynthesis. This whole slide is about the biosynthesis of the peptidoglycan layer, and I have made a video over it, and I have explained each and every step in detail. Okay, So again, I'm telling you, watch that video for just to provide a background. Okay, but now I will discuss that how antibiotics stops the different steps of the in the biosynthesis of the peptidoglycan. Yeah. Our first antibiotic of this lecture is the cycloserine. Okay, this is very much best cell wall inhibitor. What does it do? First of all, keep this thing in mind that this is a single unit. This is the this is a single unit of a peptidoglycan layer. First, a single unit should be synthesized. After that, it should be bonded to the remaining peptidoglycan layer. Okay, so this is a single unit. And just look here to this portion. This portion is very much important for this structure because I will discuss that why this is very important. But first of all, keep this thing in mind that this L-alanine should be converted into D-alanine so that it should be bonded to the fourth amino acid. Okay, this is the fourth one and this is the fifth one. So the presence of this fifth amino acid that is bonded to the fourth amino acid is very much important. Why it is important, I will now show you in a minute. But first of all, just know about the real mechanism of the cycloserine. So this L-alanine should be converted into D-alanine by an enzyme that is known as alanine racemase. Okay, so what does these cycloserine do? These cycloserine binds to the alanine racemase. Okay, this AR is the alanine racemase. It is bind to the active site, so allosterically it binds to the alanine racemase. And what does it do? It blocks the activity of the alanine racemase by what and after that this L-alanine cannot be converted into D-alanine. So if there is no D-alanine in the cytoplasm, so this to this fourth amino acid we cannot bind this fifth amino acid because we are not having it. And if we are not having the fifth amino acid, so the biosynthesis of the peptidoglycan layer cannot proceed. Okay, and then we will having little amount of peptidoglycan layer. Okay, so keep this mechanism in mind. Secondly, it also, if we are having D-alanine in our cytoplasm, in the bacterial cytoplasm, so what does it do? It binds with the help of the alanine ligase. Alanine ligase, an enzyme that bind the D-alanine to this fourth amino acid that is also known as D-alanine. Okay, so what does this cycloserine antibiotics do? These antibiotics, these cycloserine also binds to the alanine ligase. Okay, and stops its activity, and then D-alanine could not be binded to the fourth amino acid, and still the structure is incomplete, and it could not proceed further, and it could not bind to the remaining peptidoglycan layer. Okay, so this was the mechanism of the cycloserine. Now just I want to share you that why, if we are not having the D-alanine, so what will happen. So I will now show you. Okay, so just look here, this is a typical structure of the peptidoglycan layer. Okay, if we take this single subunit, if we take this single subunit, and here is the fifth amino acid. Okay, keep this thing in mind as this is also the D-alanine. Okay, and this is also the D-alanine. Here the fifth amino acid is not shown, but it is present. Now just look here, if we are having this structure, so these look to these bonds, look to these bonds. These are known as pentaglycine interbridge. Okay, and these are very much important because these bonds binds the layers of the peptidoglycan layer to each other. Okay, just suppose this is the first layer, this is the second layer, so these two layers are binded to each other. Just look here to the curve. Okay, so if we are not having the fifth amino acid D-alanine, so the enzyme cannot act upon it, and it will not bind the two layers to each other. Okay, if we are not having the D-alanine, the fifth D-alanine, so the enzyme that is used in this reaction in the binding of these two layers cannot act, and these two layers cannot be formed. So that's why cycloserines are best antibiotics. Okay, so this was the mechanism of the cycloserine. Okay, just look here again. This is a slide about it. Just look here. This is the fourth and the fifth D-alanine. So if this D-alanine is not present, so these are the enzyme that is involved in the binding of this. This is the that interbridge. Okay. So these, and this is the first layer and this is the second layer. So these two layers cannot be binded to each other to this bond if this fifth D-alanine is not present. And one thing, keep this in mind, that when the bond is formed, so then this fifth D-alanine is removed. Okay, so this also keep this concept in your mind. So if there is no D-alanine, this bond cannot be formed by the enzyme, and we will have very weak structure of the peptidoglycan layer because the layers are not binded to each other. Okay, so keep this concept in your mind. Okay, these are cycloserine tablets, and you can see them in the pharmacist lab. This is the structure of the cycloserine, and these are the tablets, the capsules, and different other type of tablets, and the basic code is the cycloserine. Okay, so if you see these drugs in the pharmacist shop, so then just remind this mechanism in your mind, that how this inhibit the growth of the peptidoglycan layer. Okay, now the second one is the vancomycin. Okay, this video should be a bit larger because I will discuss all the antibiotics that are used in the cell wall inhibition. So this is the vancomycin. What does vancomycin do? This is the unit. This is a single when the single subunit of the peptidoglycan layer is formed, so it should be bind. It should bind to the existing peptidoglycan layer. Okay, it should bind to the existing peptidoglycan layer. Okay, I will show you the picture of it. Okay, for example, we are having this unit. Okay, we are having this unit and it is now formed. So what will happen? It should bind to the remaining peptidoglycan layer. Okay, peptidoglycan, this remaining units, by a bond that is known as glycosidic bond. Okay, and so what does vancomycin do? It does not allow the binding of the single unit to the remaining peptidoglycan layer. Okay, the enzymes that are involved in the formation of glycosidic bonds, those vancomycin binds to that enzyme, and what does it do? It does not allow the joining of the single unit to the remaining peptidoglycan layer. Okay, so this is what does it do? It does not allow it, and after that it could not add units to the remaining peptidoglycan layer, and it could not increase in size. And what will happen if the bacteria is not having sufficient peptidoglycan? So it could not withstand the adverse condition. Okay, so keep this thing in mind. This is this, and these are the tablets that are used in the when in the that are present in the pharmacy lab. Okay, so keep this mechanism in your mind. Now the third one is the becitracine. Becitracine is also an antibiotic that is used against that is an cell wall inhibitor. Okay, as I have mentioned in the previous videos, bactoprenol. This bactoprenol is a carrier protein that carries the repeat unit of the peptidoglycan. It carries this unit. Okay, if I want to show you, I have discussed the real mechanism of it in the previous video. When this unit is formed, the bactoprenol acts as a carrier protein, and it carries the repeat unit from the cytoplasm to the periplasmic space. Okay, just look here. This is the mechanism. It carries it across the plasma membrane, and after that, when it carries it into the periplasmic space, so then it leaves it. Okay, then it leaves it. After that, what really happen? It have 2-phosphate, and in order to again get its original place, to get another repeat unit, it has to be dephosphorylated. Okay, it has to lose this phosphate. It has to lose this phosphate just to get access to the cytoplasm. So what does this bacitracine do? It does not allow the bactoprenol to be dephosphorylated, and if it is not dephosphorylated, it could not move across the membrane, okay, and it could not get inside the cytoplasm. And if we are not having bactoprenol, so these units cannot be transported across the plasma membrane, and it will remain in the cytoplasm. And when it is remain in the cytoplasm, so we will not have we will be not having sufficient subunits, okay, we will not be having different sufficient repeat units, and there will be no addition of the repeat units to the peptidoglycan layer, and the bacteria will not be having sufficient peptidoglycan, and it will die, okay. So this is the action of bacitracine, okay. And keep this thing in mind that becitracine is a bit toxic, and it should be used as an ointment, okay, over the skin. And in rare cases it could be ingested, okay, if it is a life threatening infection, so it could be ingested, although it is having very much side effects, okay. If I want to show you the different ointments of becitracine, so it is present in the pharmacy lab, pharmacy shop, and this is the structure of the becitracine, okay. So it is mostly used as an ointment because it has very much side effects, okay. Now the fourth one is the penicillins and monobactam, penicillin, monobactam, cephalosporin, and carbapenems, okay. These all have the same activity, okay. These all are having the same activity. And what are their activity? As I have mentioned in the previous minutes of these videos, of this video, that when there are the layers of the peptidoglycan layer should be bonded to each other, okay. It should be bonded to each other in order to provide integrity and strength to the peptidoglycan layer. So these layers should be bonded to each other, and this bond and the formation of this bond is basically known as transpeptidation, okay. This is known as transpeptidation. So what does these penicillins and cephalosporins and carbapenems do? It does not allow the binding of the two layers, okay. What does they do? Here an enzyme is involved that is known as transpeptidase, okay. If I write TP, so it is known as transpeptidase, and it is involved in the transpeptidation reaction. What does it do? The COOH of D-alanine is get binded to the NH2 of the diaminopamelic acid, okay, and it forms the peptide bond. It forms a peptide bond, just like this peptide bond. So these antibiotic does not allow the transpeptidation reaction, okay. So and just look here, it is also a transpeptidation reaction, and it is also a transpeptidation reaction, but here is a bit difference because here direct transpeptidation occur, and here a pentaglycine chain is attached, okay. So I have discussed the mechanism of it, that what does this means, but now I'm just discussing that these monobactams, this penicillin, this cephalosporin, and carbapenems does not allow the enzyme that is involved in the transpeptidation reaction, okay. Further, if I proceed, so just look here to this D-alanine and D-alanine, okay. Wait a minute, okay. I want to show you. Just look here. This is a penicillin, and this is the D-alanine and D-alanine structure. Okay. So just look here. Just look here to this structure. This is known as beta-lactam ring of penicillin. So it performs that function. What does it do? And just look here. It is a bit. There is a very much similarity in between them. So the enzyme. What does the transpeptidase enzyme do? It recognizes this site of the, for example, if this is the enzyme, okay, this is the enzyme transpeptidase, okay. So what does it do? It recognizes this part of the D-alanine, and what does it do? It binds to it and it starts the transpeptidation reaction. Okay, so if we introduce penicillin to it, so penicillin also have a similar structure to this D-alanine. Okay, this structure. So what does it do? By it directly binds, it has a high affinity to bind to this enzyme, and by binding to this enzyme cannot bind to this, and it could not perform the transpeptidation reaction. So this is the real mechanism of it. And this monobactam, if I want to show you, this is the penicillin. It consists of a this beta lactam ring. Okay, and these all are the side chains. So it also acts as a transpeptidase inhibitors. It is also, these are the cephalosporin, so it is also having a beta lactam ring. That's why just because of this presence of the beta lactam ring, these all perform same activity, but the side chains are a bit different from each other. Okay, if we look here to the carbapenem, okay, these are the these are also included in the cephalosporin group, these injections and tablets. Okay, so if you see this in the pharmacy shop, so you can easily identify that what is the action of cephalosporin. These are monobactam. Just look here, it is also having a the beta lactam ring, so it is also acting as a transpeptidase inhibitors. Now these are the medicine, and this is imipenem, and it is also very much a good antibiotic, and it is also transpeptidase inhibitors. Okay, sorry, these are also known as transpeptidase inhibitor. So this was all about that how these antibiotics stops the biosynthesis of the peptidoglycan layer. Okay, again I'm telling you, if these two layers are not bonded to each other, so the peptidoglycan layer will be very much weak, and it can be easily destroyed by the osmotic pressure and different other external condition. Okay, so hope you have understand about the antibiotics, that how they inhibit the biosynthesis of the peptidoglycan layer. If you have any question related to these antibiotics, so you can ask me in the comment box, I will answer you there. Okay, and if you have understand this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.